Hello, everyone, and welcome to today's Third Thursday webinar from the Michael J. Fox Foundation. I'm Dave Iverson, contributing editor at the Foundation and a moderator for this continuing series of webinars where we look into each month a topic of particular interest to those of us in the Parkinson's community. Today we're going to be taking up a particularly interesting topic, I think, drug repurposing, how a drug that's been approved for use in one disease may also have additional purpose for another disease. We'll be talking about a wide variety of ramifications to that concept in today's webinar. You see a few tips on the screen in case you run into any technical problems, so take a look at those as we're getting ready to go. And if you are having any difficulties, you can message, send in an email, and we'll do our best to try to problem solve some of those issues as well. As always today, you'll be able to submit your questions throughout the hour. There's a little Q&A box near the middle of the screen, so you can type your questions in there, and we'll do our best to get to as many of those questions as we can throughout our hour. And as always, we're also providing the slides today for download purposes. There's a resource list box on the screen, and so just click on that, and then the document will open up, and you can save that or print that later. All right, so let's get started with the topic that we're going to be exploring today, which again is drug repurposing. We're going to talk some about what that means. Why is it that a drug that's already been approved for one disease might have applicability for another disease, including Parkinson's? Talk some about what goes into making those selections, what makes a drug a good candidate for drug repurposing. And then the testing process. After all, if a drug has already been approved for a particular disease, why does additional testing need to take place in a new patient population? So we'll explore that. And of course, of probably most interest to everyone on the webinar today is what are some of the repurposed drug candidates for Parkinson's disease, and where do we stand right now in terms of what we're learning about those drugs and when they might be available for patient use. Joining us today in our webinar is Dr. Sabrina Paganoni. Dr. Paganoni is an assistant professor at Harvard Medical School, also at Massachusetts General Hospital, where she's also engaged in studying one of the drugs that we're going to be talking about today. Dr. Paganoni, thanks very much for being part of our program today.
Thank you. It's a pleasure.
We're happy to have you join us. And joining us as well is Dr. Todd Scherer. Todd is the CEO of the Michael J. Fox Foundation, also has a long background in conducting research himself, and is very engaged in this overall concept of drug repurposing in Parkinson's. Todd, thanks for being part of our discussion too.
Thanks, Dave.
All right, let's get started with our overall concept, and let's begin with just providing as good and precise a definition as we can of the concept of drug repurposing. So Todd, start us out on what exactly we mean when we talk about repurposing, or sometimes people use the phrase repositioning a drug.
Yeah, I think in the case for the conversation today, we're focusing on the concept of drug repurposing, which is the idea that there are drugs or treatments that are already being used in the human population for treating one disease, and then looking to see if, based on our understanding of that drug or the mechanisms of that drug, could it in fact have an impact and benefit in a different disease. So this is something that has been explored in many areas and has been a concept for some time. But the opportunity here is that there is a drug that has already passed through all the stages of drug development and clinical testing, so it's out there on the market being used already by people. And can we find additional uses for that drug to benefit other individuals who might be suffering from another disease?
So let's talk then some about what some of these possibilities are and how we go about choosing them. One of the things that we see on the slide, Dr. Paganoni, is that we observe something in the overall population through an epidemiological study, for example, that makes a drug interesting to consider. And one of those is a drug that we're going to be talking about a little bit more in detail later on that you've done some studying, which is Inosine, which is a supplement that boosts urate levels in the blood. And what is it that we've noticed about those who have higher urate levels and that relationship to neurodegenerative disease that makes it an interesting idea to explore?
Yes, this is a great point and actually quite fascinating story for Parkinson's disease and potentially for other neurologic conditions. Some researchers who are working on Parkinson noted that people with high uric acid levels were at lower risk of developing Parkinson's in the first place. And then if they happen to have Parkinson, but also high uric acid levels, they would actually do better long term. And the good news is that there is a drug that is available to increase people's uric acid levels. So from this very simple observation in people who have the disease, we are now transitioning to the possibility of testing the drug that was already available to modify, hopefully, the course of Parkinson's. And I would say that that could be a general theme that could be also broadened and expanded to other neurologic conditions. As a matter of fact, because we learned from Parkinson that high uric acid levels are good, at least when you observe the population. We are trying to see if that same concept can be applicable to other diseases. Actually, that's something I'm working on right now.
Right, you have a particular interest in seeing whether or not those high uric acid levels might be helpful for people who are contending with ALS. And we'll talk more about what we're learning so far about inosine in a little bit. Todd, another example that we're going to be talking about in some detail later is a drug called isradipine, which is a high blood pressure drug. And what's interesting about isradipine is that it combines two possibilities for making it a good candidate. One is the observational trends that we've noticed about people who take this drug and how that relates to Parkinson's. But it also gets at the second bullet point we see on the screen, which is it tells us something perhaps about the fundamental underlying biology of the disease. Tell us a little bit about that and why it's an interesting candidate too.
Yeah, so one of the things that's interesting when we think about drug repurposing and how to select some of the compounds to be tested does relate to evidence we can get from epidemiology, which is really the study of people and tracking those people. When a drug is approved and being used to treat a certain disease or certain symptom, there are people with other diseases that may have that symptom and may wind up taking that drug because we're not all segmented just by one condition. So for example, in this case where you're looking at a cardiovascular drug treating high blood pressure, there are people with Parkinson's that have high blood pressure and are treated with blood pressure medication. As an example, so you can learn a lot of information, through epidemiology studies to see what's happening in those people. In this case, with isradipine, there was some evidence from epidemiological studies that suggested that individuals who had taken this type of high blood pressure medication had a lower risk for developing Parkinson's later in life after the exposure to the drug. And that was one sort of area of evidence that came from studies of human populations. There's also a quite compelling rationale for testing isradipine or the class of compounds that isradipine belongs to that came out of the laboratory and research that was led by an investigator at Northwestern named Jim Surmeyer. The way that isradipine works to modulate the function of the cardiovascular system is by targeting the ability of calcium to get in and out of the muscle cell. And that flow of calcium in and out of the cell controls how the muscle functions. Interestingly, that same process of sort of monitoring and modulating the flow of calcium in and out of a brain cell has an important role in the survival of that cell. So what this research had indicated is that the same mechanism through which isradipine was working to impact blood pressure was also found in animal studies and laboratory studies to have a protective effect for brain cells. So you now had really two pieces of evidence around this compound, one coming from these human studies suggesting an increased risk of developing Parkinson's from people who had been treated with this medication, and now quite compelling scientific laboratory evidence also suggesting that we may even understand the mechanism through which this effect may be happening in humans. So I think combining those two pieces of evidence really highlighted isradipine as a candidate for drug repurposing into Parkinson's patients.
And so, Todd, is the bottom line here in these particular examples that we're discussing so far, it's not so much the similarity in disease. It's not like we're trying to find necessarily a link between hypertension and Parkinson's. Later we'll talk about a diabetes drug, for example, and how it may affect Parkinson's. It's not so much disease similarity that's important as it is a shared common denominator somewhere in the underlying biology.
Well, I think there's two things that have to come together. There are certainly overlaps in the underlying pathological mechanisms or the underlying biological changes that lead to different types of disease in terms of what might be happening at the cellular level or the physiological level. And then you're overlaying that with the mechanism that is targeted by a certain medication. So you could have diseases that seem to have absolutely no clinical relation, or the clinical symptoms are not closely related, but the underlying biological changes could have overlap, so that the way that the drugs are working could have this dual benefit or multiple benefits.
And that's what, and as in the case of the calcium channel connection between how that affects hypertension, the heart, and then also how it may affect the preservation of dopamine neurons. Take up one other example of what we're looking for when we look for a drug to repurpose, and that has to do with another aspect of selection criteria. And Dr. Paganoni, maybe you can describe this. We have in parentheses in that third bullet point, can get into the brain. And there's something we know called the blood-brain barrier that's there for a purpose that makes it difficult to get certain things into the brain. In this case, though, we want to be able to get the drug we're talking about into the brain. So is that one of the key criteria to assess? There's not much good in pursuing a drug that we think might be useful unless we know that drug can get into the brain?
Yes, this is really a critical component of drug development for Parkinson and for all other neurologic diseases. So there is something called the blood-brain barrier that prevents certain molecules from reaching the brain. And that's actually good under normal circumstances. But for drug development, we need to make sure that the molecules that we are using can actually successfully get into the brain. And that's one of the reasons why we need to do a lot of testing and validation in the lab to make sure that the drugs that we select for repurposing actually have a high likelihood of success. Because you can imagine even if you have a great candidate that doesn't get to the brain, obviously that's not going to be effective. So, again, all the things that have already been discussed are important to really select the right candidate to bring forward so that we use everyone's time and patient participation as efficiently as possible to maximize the impact of our trials.
So let's take up the next portion of this challenge then of getting a repurposed drug under both consideration and testing for how well it might contend with something like Parkinson's. And that's the testing process that has to go on. I want to draw everyone's attention for a moment to the kind of footnote at the bottom of the page, which is the whole reason why this idea is so appealing, is that this drug, after all, is already gone through certain approval processes and has been determined to be something that has an effectiveness and is safe for people to take by the FDA. So that means we can get it into people's hands potentially faster because it's already gone through a certain level of testing. But there's still more testing that needs to be done, right? So Todd, take us through kind of the points of this, because even though a drug, let's say a drug like isradipine that's been found to be safe to use in treating hypertension, that doesn't necessarily mean it will be safe in Parkinson's. So what are some of the questions, I guess, we have to ask when we're taking a drug that we know is safe to use in one condition? What is it that we need to find out about its safety possibilities or concerns in a condition like PD?
Right. So even though the drug is already on the market, which means it's, as you mentioned, past the regulatory approval and testing, depending on how that drug has been used in the human population, there's likely going to be a requirement to still at least do some additional safety evaluations as you're transitioning it now to testing in Parkinson's patients. And the amount of safety testing that will have to be done will vary on sort of what's known about that compound or that drug or treatment already. But some of the things that are considered in developing these types of therapies, we'll look at initially safety. So for one area, for example, that we would need to look at is there anything now unique about the biology of Parkinson's disease that could identify new or previously unexplored safety issues with this particular compound. And I can give one example in this area would be when you're looking for neuropsychiatric treatments in Parkinson's disease, particularly related to symptoms related to hallucinations. Most of the current treatments for hallucinations in the general population who have the symptom work by blocking dopamine signaling in the brain. So they're dopamine blockers. Clearly, it would not be straightforward to then expose Parkinson's patients who already have a deficit in dopamine in their brain and who are taking dopamine enhancers for their treatment to now give those individuals dopamine blockers, because the biology of Parkinson's would not be sort of encouraging of that type of treatment. So we need to sort of look at what we know about the drug and the disease to see are there additional safety aspects that we need to look at. Additionally, the population of individuals who have been exposed to the treatment may be different. So there could be a drug, for example, that has been tested only in younger people, and now we're trying to repurpose it into a population that is older. So the drug may never have been, people who are older may never have been exposed to that drug before. So we still need to then explore to see, are there any additional safety issues in this different demographic or type of population. The third point, which I got to a little bit with the example around the antipsychotic medication, is that we also have to be careful that the new medication or the repurposed medication doesn't interfere with the current treatment of the disease in an unexpected way. So that's an additional type of safety aspect that would have to be looked at. So and the last point around safety has to do with really examining the dose of the repurposed drug. A lot of the existing experience with that drug would be based on the dose that is effective for its intended and current use in the current disease or symptoms that it's currently being used to treat. If the data from the laboratory or other data is now suggesting that in the Parkinson's population we need to use additional doses or a higher dose to get the effect we want, there may be a need to explore more of the safety aspects of now using this higher dose of the drug, which is not in line with what maybe had been tested before. So again, there's a lot of acceleration we could have. We've already done a lot of preclinical testing of the safety, and there is human data that could be looked at, but there could still be the need to do some of these additional studies based on kind of the history and experience with the molecule and then related to the new disease in which it's getting repurposed into.
And Todd, would one of the reasons why a higher level might be necessary relate back to what we were talking about a moment ago with Dr. Paganoni about the blood-brain barrier, that you might need that much more in order to get this drug into the brain, and we really don't know yet whether or not that amount of drug might have other side effects or safety concerns.
Yeah, that could be one of the reasons that you need a higher dose, would be that many drugs, particularly for a disease or symptom that is not neurologically based, dependent on brain exposure, a lot of drugs are not then selected for their ability to get into the brain. In some ways, sometimes drugs are selected that can't get into the brain because it may just open up additional safety issues if it's not necessary for the effectiveness of that drug. So one reason for needing a different dose could be getting it into the brain. It could also be that where you are on sort of the continuum of the biological process that you're trying to impact, you might need a higher dose in one disease than the other. The last, related to dose, another point to be made is also how often and the duration over which people may need to take the repurposed drug. So it could be that a drug is approved for a very acute use, an acute symptom, acute exposure to a person, and in that context there's great safety data, but in trying to repurpose and transition that drug now to treat a disease like Parkinson's, where perhaps there would be more of a chronic, longer-term treatment with this drug, again, that may open up new issues that need to be explored, because it could be that people have never been exposed to the medication for that duration of time that we're now intending for Parkinson's.
And I guess related to this, Dr. Paganoni, would just be to make the point that, of course, any drug, even if it's been approved by the FDA for usage, any drug has certain side effects. We're all familiar with the side effect listings that we hear about in advertisements or read on the back of a medicine packaging. And for example, in the drug that you're interested in and that are studying, Inosine, which boosts urate levels, too much urate could lead to gout, right? So we've gotten a couple of questions that have already come in this morning from people saying, well, wait a minute, if I take a lot of that, wouldn't I get gout? So there's always a bit of balancing that has to be assessed in these questions, especially perhaps I would think if we're having to maybe even give it at higher levels than was normally intended. Would that be right?
That's absolutely correct. And that's why the clinical trial process is still essential. It's going to go faster when we can repurpose drugs, but we cannot skip that step, and we cannot just recommend using drugs off-label or for indications that are not well established yet, because again, there are potential safety concerns, and it's everything in medicine is a balancing act. So you provide benefit, but you also accept certain risks. And whether you want to accept that or not depends on very specific data that is derived by from clinical trials, so that we can actually inform patients about benefits, alternatives, complications, and people can make an informed decision. So, for example, even nutritional supplements such as Inosine can have side effects such as gout, as you discussed. So that's why it's very important that the Parkinson researchers are studying the drug in a way that's very systematic and very well organized with a lot of safety considerations, so that then we can tell patients, this is the benefit you may expect and this is the risk you are taking. Hopefully it's a low risk, and hopefully then you can balance it and make an informed decision. But again, we have to study and provide data so that everyone is on the same page and everyone can make the best decisions for their health.
Let me follow up, Dr. Paganoni, with one thing you said, which I'm really glad you raised, and it's an important point, and Patchera, you may want to comment on this as well, and that's you mentioned the use of some of these drugs off-label, meaning that someone could use this drug for a purpose other than its intended before it's gone through necessarily all these safety testing. For example, Inosine in particular is an interesting example because it is a nutritional supplement. So anybody with Parkinson's could walk into a store and get it. Or you could find a doctor who might be willing to prescribe you Isradipine, a blood pressure drug, just because we think it might be interesting. What are the concerns that you have about that as a researcher? Is it because someone then is sort of jumping to an answer before we've gone through all the safety? What are some of the trials what are some of the potential problems with that sort of off-label use, as it's often called?
There are several problems. I mean, the most obvious is, again, the potential for side effects when really we haven't established a benefit. And then, one may say, well, I understand and I want to take the risk, but then you also need to consider that there's going to be interactions with other drugs. And fortunately, there are a few options for Parkinson's, and people really need to be aware of potential interactions with other medications, because the last thing you want is to have a long list of supplements or off-label treatments that are of unclear benefit as of right now, and you don't want people to, take a number of them where they could actually lead to potential interactions. So really, it's very important to, for the scientific community to be engaged with patients and researchers and really, have a clear understanding of the goals of each treatment, again, for everyone's benefit.
Great, thank you. All right, let's get to our kind of list of candidates here. These are all drugs that are under study at this point in terms of their possibilities for Parkinson's disease. And we've talked about a couple of them already, isradipine and inosine in particular. And we may not be able to talk about all of these in detail, but of course this is, I'm sure, sort of the heart of what people are interested in most, what's out there that might have some benefit. And let's go ahead and start at the top. Amantadine is a drug, as it says, for flu. It's been around for a long time. And now it's almost all the way to the end of the line, we see on the chart there, under FDA review as a treatment for dyskinesia. What is it that makes amantadine, I guess, something that we think might be effective in helping people contend with dyskinesia, the uncontrolled movements that are often associated with long-term use of carbidopa-levodopa?
So amantadine is an interesting example to talk about, because while we're referring here to a new kind of amantadine product that is up for FDA review, amantadine is actually an example of a success story of repurposing, in that it was originally developed as a flu drug and has been used for many years now as one of the treatments for Parkinson's disease. It does also, it's a good example to highlight because it is the current amantadine that's used is also marked by some side effects as well that have to be taken into account in terms of whether it's utilized in which Parkinson's patients it's utilized. The exact scientific, underlying biological mechanism through which amantadine works is still not completely understood. It's believed to perhaps involve some of the circuitry in the brain that gets impacted by the disease. But a lot of the original use of this came from medical and clinical observations of patients that were taking amantadine who had Parkinson's disease. So it comes from that human data. The new formulation of amantadine that's being developed is really around having different exposures, more of a controlled exposure than the current drug, and that is up for review by the FDA this summer. So pending that review, we may have more aspects of this drug available for patients.
And the other drugs that we have on the screen, amantadine is really dealing with a particular symptom, not so much the progression of Parkinson's, but a symptom that's often related to extended use of a particular drug for Parkinson's, the drug that almost all of us take, carbidopa-levodopa. These other things that are on the screen, though, are also exciting because they may have some effect on slowing the progression of the disease. That's what makes them particularly interesting. So Dr. Paganoni, bring us up to date on where we are now with inosine. We see on the graph here that you're from phase 2 now entering into phase 3. And you still want people, you're still recruiting people to participate in this trial. Can you give us a bit of an update on what we've learned so far about inosine and its possibilities, whether that's in Parkinson's, or I know you have a particular interest in its possibilities for ALS as well?
Yeah, so inosine is a nutritional supplement. And when you take it, your uric acid levels will go up. Now, again, it can have side effects, but we're trying to titrate that to certain levels where we think we're going to have the maximum impact in terms of benefits with respect to disease progression while minimizing side effects. And this is an antioxidant compound, and oxidative stress seems to be a common biological mechanism across several neurologic diseases. As a matter of fact, it may be something that's important for both Parkinson's and ALS, as you suggested. So this compound could actually provide antioxidant protection for more than one disease, and that's what's really fascinating. And we learned that, again, from studies in people, but also studies in lab models. And again, this is something that could really move from the lab to the clinic relatively quickly because it's a repurposed drug. So the phase 2 study was completed a while ago, and now the current trial for Parkinson's disease is in phase 3. It's still enrolling. It's enrolling early patients and people who have recently been diagnosed with Parkinson's, and the idea is to see if actually can slow down progression of the disease. And in ALS, we're actually learning from Parkinson. So this is a beautiful example of a synergy and really a collaboration across different disciplines. The phase 3 trial of Inosine for Parkinson is led by Dr. Michael Schwarzschild, and it's actually open for enrollment at several locations in the United States. And similarly for ALS, where we are doing a smaller study, again, learning from the protocol from Parkinson. And again, we're open for that as well.
And Dr. Paganoni, we also have a request that just came in asking you to sort of speak up a little bit.
Oh,
so if you can add to your voice level as we go forward, that'd be great too. But thank you, and thanks for that update on where things stand. It's I think a really interesting and a good example of how we can learn from each other, that this cuts across not only ALS and Parkinson's, but there's also interest in it in multiple sclerosis and Huntington's disease as well. So I think it makes it a particularly interesting drug. Toucher, say something about the diabetes drug Xenotide, if you would. We haven't talked about that yet, and this is another interesting example of, you think, diabetes, Parkinson's, what in the world do they have in common? But what they have in common, I think, is particularly interesting. So maybe you could describe a little bit about what's intriguing about it and then where we stand with what we're learning so far.
Yeah, this is another example that really came from some interesting laboratory research that was looking at, again, two disease processes that at face value would have thought were unrelated. But actually, one of the mechanisms that is involved in some of the deficits in diabetes that is targeted not only by exenatide but by a class of compounds has been found to also play a role in neuronal survival, or the survival of brain cells. And then some laboratory studies were done to show that these members of this same class of drugs that are already developed for diabetes were protective in the model systems that were used in laboratories. So again, this then, because these drugs were available, allowed a more rapid progression into clinical trials. And we are expecting the results of sort of this next exenatide trial to be announced this summer, which was the phase 2 trial. And like not only exenatide, like I mentioned, there's an entire class of available drugs that are targeting the same mechanism. So we have an opportunity to not only look at that biological mechanism, but then over to see which of these compounds may be most effective in the context of Parkinson's in terms of dosing and blood-brain barrier exposure.
And Todd, let me just stay with you and ask you to also give an update on nilotinib, which is the cancer drug that has received a lot of attention in the last year or two. There's some studies that are just underway now through Georgetown University. Fox Foundation is also engaged with the Parkinson Study Group in another trial that will be launching soon. Again, fascinating example. Diseases that on the surface seem differently, but there are mechanisms that make it appealing. What do we know so far about that? And again, this is these are our trials along with Inosine, where people will soon be able to enroll on them if they visit our Fox Trial Finder website. But walk us through a little bit of what we've learned about nilotinib so far.
Yeah, this is another example of a drug that got nominated out of some laboratory research, and in this case, the nilotinib is from a class of drugs that is used to treat a chronic form of leukemia, and targets a certain cellular pathway that's involved in the survival of the cells impacted in leukemia. And researchers in a number of different laboratories then uncovered that this same biological pathway was involved in how the brain cells handle the protein alpha-synuclein. Alpha-synuclein, as a reminder, is a protein that is aggregated in the cells, brain cells of people with Parkinson's. So there was some evidence from the laboratory that targeting this mechanism could help the cells to degrade or get rid of this aggregated alpha-synuclein, and by targeting it with a drug like nilotinib and drugs from this class. And based on that data, there was an initial small study that was, the results were announced last year. There are issues that have to be explored as a cancer drug to really look at safety. So this is an example where just because the drug has gone through all the approvals, there are known side effects of drugs, and particularly cancer drugs that can have known side effects. So this is something that's being explored in the current studies, as well as looking for whether there's a benefit for patients.
All right, let's go back to our next slide.
Dave,
yeah, go ahead.
There was one other point I wanted to make, just looking at this portfolio, and it's something I think for us to be cognizant of as we talk about drug repurposing. So one of the advantages, again, of drug repurposing is that these are drugs that are already on the market, being developed, being sold. But one of the challenges is that depending on where those drugs are in terms of their life cycle, are they, for example, off patent because they've been around for so long, like Inosine and Isradipine sort of fit this category. In that case, it takes sort of a unique funding model to also move these drugs forward, because there isn't necessarily the same drug pharmaceutical company profit incentive when you have a patented compound. So in the case of Isradipine and Inosine, those have moved forward. Exenditide, the same, and now nilotinib have moved forward with unique funding models of foundation, Fox Foundation, National Institute of Health government funding. These are funders that don't necessarily have that same profit incentive, but these trials are extremely expensive, so it does take sort of a unique approach of how to support and move these projects forward from a financial perspective, because there is not that same for-profit motive necessarily, given where these compounds may be in their patent lives.
No, it's a great point, Todd. Thank you for raising it. It really does underline the key role that both foundations and federal funding through the National Institutes of Health in particular play, because we can't just rely on pharmaceutical companies to do this. If there's no profit incentive, they're not necessarily going to. And if anyone ever wondered why it's important to support foundations or federal funding, this is a great additional reason to make clear why that can be so key. All right, we're going to put up on the screen now just kind of a sort of some overall summary points about the topic we're going to be that we've been discussing. And as we do so, we want to start taking all of your questions. There have been lots of really good questions coming in, and we want to start getting to all of those at this point. Let me start with this one, Taksha, that came in from someone who says that they've been participating in an Isradipine trial for the last 2 years. And they're concerned about what's going to happen after the trial is done, because they say this person says has done well. They think Isradipine may be one of the reasons why this person is doing well. Indicates that her UPDRS scores have stayed the same while she's been taking this drug. And the concern is whether or not, once the trial is done, and then there's this longer FDA review, what happens then for people who have participated in trials? Can they still get access to the drug, or do they then just have to sort of sit on their hands until the final FDA approval is finished?
Yeah, so this is an interesting question as it comes to the repurposed drugs, because, so again, these drugs are available in terms of that they're on the market now. So I think depending on the results of the trial, as we get more and more, it depends on the results, of course. If the results are positive and the medical community now has better understanding of how to use the drug in a disease with good rationale, there could, it's an unclear exactly what are all the gating steps in terms of FDA approval for the use of the drug. That would give it a on-label use for Parkinson's. But there is always the potential for off-label use of these drugs, particularly if it's based in strong data. So I'm not sure that you would have to necessarily wait indefinitely or for a prolonged period of time. The one caveat I would raise, though, in terms of how the question was even framed, is that if the use of this drug is through the course of a trial, we should remind everyone that it's a blinded trial. So that means some of the people actually got Inosine and some got the placebo, which means, did not get a new medication. And both in a well-designed trial, both the investigator and the participant should be blinded, double-blind trials. So that's why it's so important to then look at the results and see, was it in fact due to the medication that the group who got the medication did better than the others? So we don't know for sure whether this person is on the experimental drug, in this case is Ratapine, or is in the placebo-treated group.
Yeah, no, that's a really important point too. We sometimes can easily forget about you don't really know whether you're getting the drug or whether you're getting a placebo. But Dr. Paganoni, Todd raises an interesting point. You were describing earlier all of the concerns that we understandably have about people using things in an off-label way, but Todd, I think, is making the point that once all the data is understood, and we really know that there's pretty legitimate data here suggesting that one of these drugs may in fact be useful for Parkinson's disease, then I'm thinking then the next possibility might be that a physician could and a patient could, in good conscience, then consider using this drug, even though it may not have gone through all the steps of FDA approval. That makes sense? And would you consider that as a physician to take that approach once all the data is in?
Yes, it makes sense if the data is positive. Obviously, if the data is strong and how strongly positive it is. And, every trial is designed for certain groups of patients, and we need to know if the results are more generalizable and, the particular situation of the patient, other medical problems that the patient may have, other drugs that the patient is on that may interact with the new drug. So based on all of these, issues, one could actually consider prescribing it off-label after the trial is positive, especially if it's strongly positive. And I would expect the FDA approval process to go faster for drugs that are repurposed if the trial is indeed strongly positive. So again, I think that there's a lot of potential for bringing drugs to market faster with this approach.
Yeah.
And Dave, I think it's also important to, in this conversation, to remember that repurposed drugs are not all created equally. We've talked on one level of things that are might be a nutritional supplement or could be a vitamin or something, all the way to the other extreme. You could have something that's known to be a highly toxic cancer drug or something on the other end. So I think it's also with the data, more data provides more informed decisions. So the more data we have from the trials, then just like was just said, then the patient and the doctor could sit down and look at the pros and cons. But there is a sort of a continuum of what's known about these medications. Some of them have been on the market and have treated thousands and thousands of individuals, and some are approved, but they're only approved for very niche or small populations of patients, and there's a lot less experience with those drugs in the human population. So it's not a sort of one-size-fits-all decision process, which is why it's so critical to continue to develop the appropriate data around each of these drugs and get more and more experience in what could be the risk-benefit ratio that should be considered in terms of using them.
Yeah, I think that's a good cautionary point to add on that. And I think with all of these things, we're kind of balancing interest and enthusiasm with reminders about proceeding with caution as well. Todd, that leads me to a next question that I'll pose for you, which has come in that has to do with the cancer drug nilotinib, which, as you were suggesting earlier, cancer drugs in particular have to be approached with caution because they are often highly toxic. So the question that came in is the dose of nilotinib that's being currently given to a leukemia patient, will that be the same dose that is tried in these upcoming Parkinson's trials?
Yeah, so my understanding is that in the current trials, the dose is lower than what is normally given for leukemia. So it'll have to then be explored whether you have, what's the safety look like and do we get the efficacy. But this is an area where the researchers have titrated the dose back for a lot of the reasons that you just raised. And now you have to, we're hoping that that lower dose will have the benefit that we want.
Okay. And Dr. Paganoni, a question for you. One person raised, well, what happens if I were to boost my inosine levels? Do I have to worry about gout? So tell us something more about that relationship and what one might want to be concerned about in that regard.
Right. There are two main concerns in terms of side effects from inosine, one of them being gout and the other one actually being kidney stones. So both are possibilities in a small proportion of patients taking inosine. Now the risk is higher if one takes a lot of inosine. So it's very important for anyone who would ever even consider this to be under medical supervision so that the levels can be strictly monitored. At this point, again, I would not recommend to taking it without being part of a clinical trial because we're still learning about the safe dose range, and that's actually one critical aspect of the trial to understand how, what are the best levels that we can target to get the maximum benefit with the smallest possible risk of side effects. So again, it's not that just taking the drug will give you, necessarily gout or kidney stones. It's all a matter of how much exposure you have, for how long, and how tightly you are checking the levels. So all of this, your physician could help you with. But again, at this time, I think it's a great time for research on inosine because again, the trial is underway and it will clarify, exactly how to take the drug, what's the impact on disease, and the sort of the safety risks that are associated with it.
Okay, I'm going to put back on the screen now our list of drugs that are currently being studied for repurposing possibilities in Parkinson's, because Todd shared the question has come up that some people aren't clear about which of these might be disease modifying, in other words, slowing down the progression of Parkinson's, and which are drugs that might actually just be focused on improving symptoms, even if they didn't slow down disease progression. And of course, something could do both. But walk us through this real quickly, if you would, Todd. We know the top one is more symptomatic, but describe what the hopes are, I guess, for each of these in terms of their symptomatic benefit versus the possibilities that they would slow down disease progression.
Yeah, so I think for the isradipine, inosine, exenatide, and nilotinib, the rationale for moving these forward has to do with targeting of the underlying disease process. So the goal or the hope is that by targeting these underlying disease processes, you could impact the progression of the disease. It raises an interesting point, like you may have been getting at or not, but I think it's interesting, which is that whether you're testing a repurposed drug or a new drug, it doesn't change sort of the challenges of doing the clinical trial and actually dissecting out whether you're having a symptomatic effect, acute symptomatic effect, versus a longer-term disease-modifying effect. So in terms of the trial designs, the trial outcome measures, the underlying biomarkers that are needed, those will all be equally required in these trials. And it gets to the point I was making before that the trial itself still needs the same resources in terms of the number of participants, the duration of evaluation, and other outcome measures that are used. So we don't really know the answer. We know why these were nominated, but it's possible that they could have symptomatic benefits, and it's also hoped that they would have disease modifying, and that would be borne out in the clinical trials.
And as you suggest, this again underlines our need and hopes as far as finding a definitive biomarker, an objective biological measure, because it's very difficult to know whether or not something is actually slowing down the progression of the disease unless we have an objective way to measure it. So the ongoing pursuit of finding those more definitive objective measures for Parkinson's and other disorders is crucial in order to be able to assess test results in the best way. While we have this list up, Toucher, let me ask you one other question that's come in, which is someone wondering whether or not any of these conditions, and Dr. Paganoni, feel free to jump in on this too, would have an effect on other conditions that are related to Parkinson's, but are different conditions. This person wondered if any of these would be helpful for multiple system atrophy, for example. There are other conditions like progressive supranuclear palsy and others that are in some ways sort of more acute forms of Parkinson's, one might say. Do we know, Toucher, whether any of these would have an effect on some of those other conditions too?
I definitely think it is possible. It all will depend again on what the underlying biological mechanism or process that the drugs are targeting. And particularly, for example, we talked about Inosine, which is targeting it's an antioxidant, so it's targeting these increased oxidative damage that can happen across neurodegenerative diseases. There's reason to believe that if it works in one, it could work across many. So I think this goes back to sort of what the underlying biology is for how these drugs may be working, and there's certainly a rationale that could be developed for those other related diseases that you just mentioned.
And Dr. Paganoni, that's of interest to you in a sense because in Inosine in particular, it seems to be something that could apply to a variety of other neurodegenerative conditions.
Yes, that's our hope, and that's what we're testing right now. Specifically for Inosine, we're testing if it's helpful for ALS, also known as Lou Gehrig's disease, which is actually a different neurologic disease. But, it's biologically possible they may help other diseases as well, especially those that are more related to Parkinson. All of these could be tested in future trials, and the hope is that, again, by using drugs that are being repurposed, trials for other diseases will be faster.
Here's an interesting question that's come in about whether or not whether there are any treatments for autoimmune diseases that might be candidates for drug repurposing in Parkinson's. Share. There's been some research recently that's raised this question of whether Parkinson's is in part an autoimmune disease, that there's something that's going on with those sticky clumps of protein that kill neurons, we think, in Parkinson's, alpha-synuclein clumps, whether or not the body's immune system mistakes that for some sort of invader and attacks, and that furthers the progression of the disease. The question is whether or not are there drugs now for current autoimmune disorders that might be candidates for drug repurposing.
Yeah, so first I just want to credit again the audience that we get for these webinars, because this is exactly what the scientific community does in sort of evaluating the potential around repurposed drugs, and there was an extremely exciting new study that just came out recently, which indicated some of the first evidence that there could be support to this hypothesis that Parkinson's could be an autoimmune disease, or there's an autoimmune component to Parkinson's. This is the exact kind of laboratory work and translational work, when you then look at some of the evidence from people with Parkinson's as well, that is used as the basis to evaluate and examine new opportunities for repurposing. And this is certainly an area in terms of the role of the immune system in Parkinson's that is gaining more and more momentum and more evidence around it as a key mechanism of the disease. So this is something that is being currently further explored to try to develop more and more information around this to see whether it could be targeted through repurposing, and also the development of new medications, and also to be looked at as a potential biomarker for Parkinson's in terms of measurements of the immune system.
Yeah, it's a fascinating question and a great question. Thank you for sending that in. Dr. Paganoni, a question about risks associated with participating in these clinical trials that are focused on drug repurposing. Questioner wants to know if there are any different risks associated with participating in those clinical trials. Since you're still recruiting for Inosine, what's your thoughts on that? In some ways, I guess I would think that the converse might be true, that because these drugs have already been approved for another disease, we know already some things about their safety, right? So would that mean participating in a clinical trial for a repurposed drug on the surface, at least, would perhaps be a safer endeavor?
Yes, I would say, I want to reassure the audience that the level of monitoring and really the attention of the investigators is really, really high for everyone who's enrolled in a clinical trial. I myself lead 4 clinical trials at the moment for ALS, and I can tell you that I spend a lot of time really, really thinking through, all the possibilities and how we can improve the safety and the well-being of all the participants. So whenever you are in a trial, you really have a team that's really working essentially on your behalf to try to really ensure the well-being of every participant. There are multiple layers of really monitoring and supervision. So yes, there are risks associated with any drug or being in any trial, but I can assure you that the way clinical trials are run and regulated really provides to the best of our abilities, really a level of protection, at least, monitoring, so that the safety of every participant is monitored pretty much in real time. So definitely there are a lot of safeguards there, and there are a lot of regulations actually that guide really the conduct of these trials.
And Todd, a very practical question about whether or not drug repurposing is one of the benefits of it, that it will lower costs, that these drugs, if they're repurposed drugs that have gone beyond patent, will they be cheaper?
I think it would depend on the particular drug. For example, nilotinib is currently a very expensive medication, but some of these drugs have gone to generic forms, so it's not only is the patent up, but are there generic forms of the drug? And in that case, the answer is definitely true, that it would be less expensive if what's being tested is a generic medication. Something like Inosine is not, expensive compared to nilotinib, which is, much more expensive.
All right, I'm going to put up our last slide, which is just our call out to participate in research. It's such a key part of our overall mission, and you can find out about ongoing trials, not only of repurposed drugs, but all Parkinson's-related drugs by visiting foxtrialfinder.org. But I want to conclude as we look at this last slide with a great question that came in from Jane, and ask you to both comment on it as a way of kind of summing up where we are. And Jane's question is, are you more excited and optimistic about a repurposed drug as a breakthrough treatment than you are for a novel drug being treated specifically for Parkinson's disease. So, Dr. Paganoni first, is this an avenue, these repurposed drugs, that you find particularly excited about making progress in combating not only Parkinson's, but the other neurodegenerative diseases that I know are of particular interest to you?
Well, I would say that the difference between the two is that when you're considering a repurposed drug, you already have more information to start with. So it's not like I'm more excited about repurposed drugs as opposed to sort of regular drugs or completely new drugs. It's just a matter of timing and opportunities. You know, what can we do to get new products to the market faster for patients with Parkinson's disease? And I think we should be open to both very new and experimental and high-risk new drugs as well as repurposed drugs that can perhaps get to the market faster.
And Todd, I'll give you the last word on this. Perhaps you could also mention in your answer as to how you view your excitement about repurposed drugs, that they're not necessarily mutually exclusive. You were telling me when we talked about this a week or so ago that you could test a drug, repurposed drug, and it might kind of get you almost there, but not quite, and then that might lead to sort of a reformulation of that drug into a novel drug for Parkinson's. So it's not necessarily one or the other.
Yeah, I think that the repurposed drugs could also provide a much faster way, because they could get to the clinic faster than a new drug, to open up a new mechanism or new avenue for research. And it could be that it's a stepping stone, so they show a benefit, and then we could build sort of on top of that to get an even better drug. And that next drug could be a new drug that's developed sort of on top of the same mechanism, but now that there's this proof of concept that this mechanism could work, there's a lot more interest to move in that area. You know, when I think about, you know, am I more excited about repurposed drugs versus new drugs, one thing I want to make sure to remind the audience that the pipeline we showed here of repurposed drugs being tested, there's an equivalent, if not larger, pipeline now of new drugs that are at similar stages of drug development for Parkinson's that are new drugs. So there's a very robust pipeline of drugs being tested in the clinic, many focused on disease progression. And when I think about, you know, people ask me all the time, sort of, what are you most excited about in the portfolio? And I try to refer back to, I love both of my children equally, and I want them both to get full scholarships to college and be very successful. So we are, you know, pushing forward on all of these. And what I'm most excited about is that all of these hypotheses for Parkinson's have now moved this far into testing. And, you know, the more chances that we have of things being tested at this level, the better chance we have that one of them will push through to the next stage.
And we will close on that hopeful note. Dr. Todd Scher, Dr. Sabrina Paganoni, thanks both so much for your participation today. Thanks to our audience, as Todd noted, some particularly insightful and thoughtful questions. We appreciate those so much too. They really help make these webinars, we hope, to be a satisfying experience for all our participants. Again, a reminder that you can